Well, hello, hello. Something I just realized is let me share my screen here. I've come to the realization that these are the number of classes that we have left. So this is the equivalent of 1 and 2 weeks. In well, 2 weeks, and including this week, that'll be 3 weeks. So that's the number of classes that we have left. Um, so be thinking about how you want to wrap up this semester. If you've fallen behind on some things, please reach out to me in person. Don't flood me with emails. Yeah, just let's talk about anything that might have occurred or that you might have missed in person, and we'll see what we can do about it. Okay. So yeah, 3 weeks left. So keep that in mind and be thinking about how you want to wrap up your semester. So where were we? We looked at the olfactory system. Um, I hope all throughout this you're keeping in mind how the initial stimuli interacts with the receptors. Whenever we look at the olfactory system, we also saw that it was actual molecules, actual odorants that come into contact with the receptors within your olfactory neurons. So basically, that's the reason why you can't smell. For example, I can't smell this glass. It's just I can't smell it. The reason being is because there are no volatile substances just coming out of the glass, being aerial, aerial coming into the air, and and then in contact with your olfactory, the receptors in your olfactory neurons. And that is why cold food doesn't smell as good as warm food. Whenever you heat up the food, you all of a sudden you start smelling that. Why is that? It's because it's hot. I mean, like that's what that's what common sense would tell you. However, the actual mechanism of it is like microwaves excite the atoms and molecules within your plate of food. Those atoms, now that they have been stimulated by this energy, they start moving. They all of a sudden they are in motion. That is what heat is a substance in motion. That's why whenever you do this, what's that? It gets warm. It's because you have produced friction, which in turn has produced motion, and that motion in turn produces heat. So back to the plate of food. The plate of food now, those molecules, they're moving. Whenever they move, they're most likely to go from like this solid passive state into for them to be aerosolized. They they become the molecules start floating in the air, and now that gives that there are more of those molecules from your food in the air, and there's more chance of them coming in contact with your olfactory receptors, and that's why you can smell warm food. That's why warm food smells better, or that's why we like warm food as opposed to cold food. So keep that in mind whenever you smell something. Whenever you smell something, it's like, oh, I'm actually the substance that I'm smelling, the plate of food, the public restroom, everything that you smell is the source, molecules from the source becoming aerosolized and then coming in contact with your mouth, with your olfactory epithelium. Sorry. All right. Same goes for whenever we're looking at whenever we were talking about the gustatory system, you have molecules from the gustatory system, molecules coming in contact with receptors within the hair cells of these gustatory cells. Once again, actual physical contact for both senses so far. Now we were looking at the visual system. So far, we've seen that it's a complex system, lots of moving parts to it. So a lot of them you will learn in the anatomy portion of this. But one of the gists of this, you have to learn them simultaneously in order for us to understand how they function altogether. But one thing that that occurs within the system is the refraction of light as it passes from the air into a denser medium. There are several mediums that it goes through. It's going to go through your cornea. It's going to go through the aqueous humor within the anterior cavity. It's going to pass through the lens. All of these substances, all of these structures have different density. That means that light is going to bend as it passes from one density into the next. And finally, it will pass the vitreous humor and reach the macula, specifically the fovea, the most central region. Even though there is a level of refraction or bending of light as photons pass through these different portions, the part where most of the refraction occurs is going to be your lens. And that's something that I guess we intuitively are aware of. I mean, like, that's the role of the lens, right? That's the role of a camera lens. That's the role of your eye lens, is to focus light within in our case, it's a retina. In the case of a camera, it's going to be a sensor or a film strip. The lens will focus the light in that particular position. It does so by changing the focal length of where that beam of light will focus. If you stare really close, we're going to see how those focal points would change, whether you're staring into the horizon or if you're staring at your hand right here. The focal points are going to be different. However, your lens is going to make sure that those focal points are going to, regardless of whether you're focusing far away or whether you're staring straight at your hand, the lens is going to thicken or slim down in order to focus that to focus that into your retina. All right, we talked about the vascular layer, and it's going to contain blood vessels, lymphatics, and the vascular layer will also produce and reabsorb aqueous humor. The iris, which is going to be that the color portion of our eyes, contains blood vessels, pupillary muscles, and pigmented cells, which are going to, in large part, give that difference in eye color that we all have. The pupillary muscles themselves are going to serve as a as a diaphragm, analogous to the lens of a camera that will determine how much light is going to pass through into the retina. I don't know if you stare into the sun, you reflexively close your eyes, even though your pupil itself will dilate. Um, we dilate as much as it can so that no excess light makes it through. But you still will reflexively close your eyes. Don't stare into don't stare into the sun. Um, but that's what happens. We have these systems that allow us to basically modulate the amount of light that makes it into the retina. The retina is sensitive. You could damage your retina if you stare into the sun for too long. Those mechanisms are going to be analogous to not only chemical in the sense that we'll see later how the mechanism works, but you will basically be photo bleaching your photoreceptors, and also you could damage them, analogous to the damage that occurs from a sunburn. All right. The ciliary body contains muscles, and this is what we are. The ciliary body contains muscles. Those muscles attach to the lens through the suspensory ligaments. So the lens is suspended by these ligaments that would either stretch it or allow it to relax, and that changes its thickness. And these suspensory ligaments are going to be attached to these ciliary muscles, these muscles that allow this modulation. The choroid itself, that layer is going to be a vascular layer. Here is the choroid layer. It's going to be interior to the sclera. And here you have the your ciliary muscles, and here are the suspensory ligaments. So let's look at, and here we have the pupillary muscles. You're going to have the constrictor pupillary muscles and dilator pupillary muscles. And they pretty much do what their name suggests. So the dilator muscles, you see the setup, how one of them is surrounding the pupil and the other one one of them kind of squeezes the pupil and the other one is radial to the pupil. The one that the constrictor muscles, they can take the shape, basically dilate in the process, closing the eye, closing the amount of light that can make it through the pupil. And the dilator muscles, whenever they contract, they will pull. Whenever they contract, they will pull the constrictor muscles and create a larger opening in the pupil. Therefore, more light can pass through. Apparently, I hear that whenever you take certain drugs or certain chemicals, that these will dilate. That should inform you of some of the pathways that are being affected. Whenever a particular medication has this effect, you should be thinking the activation of your sympathetic system, right? Because this is what happens whenever we activate our sympathetic system. Basically, our eyes, we feel under threat and our eyes go, where is the threat coming from? Your pupils dilate to be able to, I don't know, see better. So that's what you should be thinking whenever you see any of these effects. Is like the activation of the sympathetic nervous system. Probably something that counters that will will counter the effects of whatever medicine we're referring to here. All right, so let's look at the retina. Contains the outer portion contains the pigmented area, and we talked about the role of that pigmented area. The inner contains the retina. The inner contains the neural part. That's where your rods and cones are going to be. Highly focused on the fovea. Here, we're going to talk about this retinal pathway where your photoreceptors communicate with bipolar cells, then ganglion cells, then optic nerve, thalamus, and cortex. These two, except for, we mentioned, except for the olfactory system, all of the systems that allow us to monitor the exterior world go through the thalamus and then into the cortex. Here we're talking about the optic disc. We also have horizontal and amacrine neurons that modify signals. So here you have a view of the exterior, the outer pigmented area, and this neural layer. This is the area that contains your rods and cones. Here are the cell bodies of your rods and cones. Here are we're going to see this, the area where the photoreceptors are going to be present, the actual receptors for the photons of light. They're present at this level. Here we have a layer of bipolar cells, or these are going to synapse with this, and then they're going to synapse with ganglion cells, and then off to the thalamus. We mentioned also horizontal and amacrine cells. You see horizontal and amacrine cells. They are there whenever you see cells that communicate with several cells of another cell population. They're there to mainly modulate, not only modulate the signal, but also kind of get a feel of what the signal, kind of integrate the signal at the level of this population. So this horizontal cell is going to it's going to produce a signal whenever it's going to tell you what these other 10 neurons are doing. So it says, so now you have basically translated the communication of 10 neurons into one neuron, the horizontal cell. And in that way, you get, this is the way that the neuronal signals are kind of being coded and interpreted, not only in a one-to-one neural communication ratio, but also as a many-to-one and a one-to-many. So several different nodes where these signals integrate and also diverge in order, and all of the combination of those different interactions is going to determine what we perceive as the actual context of all of those photons, all of those interactions of neurons firing. It's extremely complex to the point that we're still, in large part, there's still a lot of mysteries about how this neuronal language, what's the complexity of this neuronal language, what's the benefit of it, some of the downfalls of it. Someday we will understand it better, and hopefully when that day comes, any maladies that we may have, they'll be treated appropriately. Right now, we're treating vast populations of. We're treating at the level of neurotransmitters and basic. For example, take um, serotonin antagonist or any of these substances that act at the level of the neurotransmitter. They don't uh, target a specific population of neurons. They target the entire function of the neurotransmitter. And those neurotransmitters, there are side effects with certain medications because that neurotransmitter is not doing just one function. It's doing many functions, not only within the nervous system, it has functions in the digestive system, vascular system, in many systems. So whenever we take medications that in somewhat in this, the target a particular neurotransmitter, we're affecting several levels, several functions. So that is why a lot of treatments for some maladies are hit and a miss because we still don't understand the brain enough to be able to determine exactly how to approach some of these treatments. Someday, hopefully, we'll be able to target specific populations of neurons, and whenever we have total understanding of what this specific population of neurons do, and we can appropriately target just this specific population of neurons, just a neurotransmitter on this specific population of neurons, then we can talk about real efficient and effective treatment for some maladies. All right, here we are, the optic disc. Boom. All of those neurons that we were talking about, they have to leave the eye somewhere, somehow. They all do so through the optic disc. This disc, it's also a place where the vascularization comes in and out of the eye. So this will not have photoreceptors. You see the photoreceptors here, they're the ones responding to light. They're not present here. And that's what it's known, that's why it's known as a blind spot. Boop. There. All right, let's look at the chambers. So we have two chambers, an anterior and a posterior chamber. These chambers are going to be separated by the lens. Sorry, we have an anterior and posterior cavity separated by the lens. The anterior cavity is further divided into anterior and posterior chamber. Just know that cavity bigger than chamber in the visual system nerd terms. These chambers are going to be separated by the iris. So here we have posterior cavity separated from the anterior cavity by the lens. And within the anterior cavity, we have an anterior chamber and a posterior chamber separated by the iris. The anterior cavity is going to be filled with aqueous humor. The posterior chamber is going to be filled with vitreous humor. The aqueous humor is going to be produced at the level of the ciliary body just through these highly vascularized areas. Once again, whenever we're creating fluids, be it urine, be it mucus, be it sweat, be it saliva, cerebrospinal fluid, any fluids, any liquids that we are creating they come from the circulation. That's where all of our liquid is. Well, that's what most of our liquid is. So they come from the circulation, and eventually they go back into the circulation. They go back into the blood vessel. And here we have these capillaries allow this formation of this aqueous fluid, and eventually the fluid itself gets drained to the canal of Schlemmel, and it goes back into the venous system. The vitreous humor is more permanent. It's more, it's denser. It's not recycled as easily as the aqueous humor. It's more permanent. So once it is formed, it basically remains there. If the formation of aqueous humor and the drainage of it does not happen in synchrony, then you have these differences in pressure that might affect your visual system. All right, here is the lens. Separates anterior and posterior cavity, and the lens will help focus, accommodate light, and it says normal acuity is 20/20. Here is an example of what happens. This is this is just us. I bet this is what's happening right now within your visual system. You're staring at something that's not too close, but that's not too far either. That's not in the horizon. So the lens is going to focus this at this point. That point is usually the retina. If you have 20/20 vision, that point should be the retina. However, if we wanna stare at this is so this is what's happening right now in your hands. It's not at its scale, so it's hard. This is what's happening right now in your eye. If you stare at a really close source, like if you stare at the way that I'm staring at my hand, it's just like a couple of inches in front of my face, then the focal point will increase, and that focuses, the focal point will increase. And if you stare at an object that's far away in the horizon, the focal point decreases. So in order with these changes in focus and focal distances, in order for the focal point to be exactly at the same place, the retina, then your lens will change shape in order to accomplish that. So here we have an example of if an object is nearby, then the lens will thicken in order to bend the light farther, because you're providing more of a thicker lens will provide far more bending of the light. It's analogous to light traveling through a cup of water, or light traveling through an entire I don't know, fishbowl. There's going to be more bending in the fish of the light that travels through the fishbowl because of the density compared than when a light. So that is how the lens is making sure that the focal point is always your retina, specifically the macula and the fovea. For distant vision, the lens is going to thin out, it's going to relax. That way, there's not going to be the necessity for much bending of the light in order for this to happen. The closer the object is to the source, the larger the focal point. So if the lens were not to thicken, then this image will probably focus over here, farther along. But due to the thickness of it, the signal, as the lens thickens and thickens, then the focal point comes closer and closer to the retina. Eventually, if the lens is thick enough, that the focal point will be along the retina. In this case, the opposite is true, but probably the focal point, you don't need to thicken the lens as much in order for the focal point to hit the retina whenever we're staring at faraway objects. These are examples of visual abnormalities, emmetropia, everything's kosher. You have myopia and hyperopia, where the focal point is before the retina, and where the hyperopia, where the focal point is after the retina. How do you fix this? Well, if it gets before the retina, you wanna expand, you wanna basically prevent this. You can almost see yourself physically opening this beam of light, just like making it, preventing it from it to focus before it hits the retina. And that's what diverging LEDs do. They open the beam of light. That way, before they reach the retina, they already diverge, and now it takes longer for these beams of light to focus. The focal length increases because we have basically opened the beam of light. In order for the focus point to be farther, in order for us to elongate, to make the focus point larger, increase this distance, basically. In order for us to screen this distance, we need to diverge or open the photons of light. That's what diverging LEDs do. If the focal point is too far away, we need to converge these beams of light to decrease the focal point. That is what converging lenses do. Rods and cones, we talk about rods sensitive to light, to dim light, and cones, they're your color sensing compound. Your color sensitive photoreceptors, and they respond to blue, green, and red. So you have a population of cones that only responds to blue light, a population of cones that only responds to red light, a population of cones that only responds to green light. If you notice a rainbow, you're basically, you have them situated so that you have a portion of the rainbow that, so the ROI, that would be the red photoreceptor, the G, that would be the green photoreceptor, and the blue, the ultraviolet area, that would be sensed by the blue photoreceptor. Let me see if I have a photo of a rainbow here. But there is an overlap in, it's not, these photoreceptors do not, the wavelengths that they respond to, they're not bucketed, they're not beamed into exclusive wavelengths. They are overlapping. So the green photoreceptor will respond to a portion of light that the red photoreceptor will respond to a wavelengths of light that the green photoreceptor also responds to. So whenever you have a beam of light in the between red and green, for example, an orange color, then depending on how many of the red photoreceptors and the green photoreceptors have been activated, that gives us a sense of the in-between colors, in between red and green and blue. So let's look at how the actual interaction between the photons of light and the photoreceptor occurs. These are your cones. These are your rods. Your cone. Oh, both of them are going to contain this. They contain an inner segment and an outer segment. This is why they call them cones. Look at the shape. And this is why they call them rods. Look at that shape. They contain within them. They contain these discs. This particular disc contains this photoreceptor molecule. This photoreceptor molecule, it contains a protein called opsin, and it contains also a compound called retinal. So this is your rhodopsin molecule, your rhodopsin photoreceptor. So whenever we're in, so I want to show you first what happens whenever whenever light. So you have your closed eye. Now you've opened your eyes. What happens whenever we open our eyes? This particular photoreceptor reaches a negative 70 millivolt. That's the resting membrane potential of neurons. That's what neurons. That's the membrane potential whenever most neurons are not doing anything. So that's whenever light hits us. So there is basically no signal being sent. Whenever we are whenever it is dark, that's whenever the membrane potential becomes more positive. It goes from negative 70 on light conditions to negative 40, so 30 millivolts more positive. Whenever we close our eyes, and you know what happens. This is because sodium channels open. What happens when sodium channels open? Sodium goes into the cell. That happens in most cells. I can't think of an exception. I don't think there is one. But whenever a sodium channel opens, the sodium rushes into the cell. This is why on dark conditions, the interior of the cell is more positive, because all of that positive sodium is going into the cell. Why is it doing this? Is because of in dark conditions, we have retinal in a cis configuration. Whenever retinal exists on a cis configuration, you have basically cGMP activating this sodium channel. cGMP level, this cycle messenger system, cyclic GMP, activating this sodium channel, opening it in the process. Whenever it opens sodium channel, then sodium from the exterior of the cell just rushes into the cell. This is in dark conditions. And that's why the cell now is at negative 40 millivolts. It's releasing neurotransmitter because this basically is threshold for neurotransmitter firing, for threshold for action potential. But what happens whenever a photon of light interacts with the retinal of this rhodopsin 4 receptor? Whenever a photon of light interacts with the retinal changes configuration. It goes from this cis configuration to a trans configuration. These configurations are cis versus trans, are relative of how hydrogen bonds are, certain bonds are exist in relation to each other, whether they exist on the same side or opposite sides of a particular bond. So cis, they both exist within the same side, and trans, they exist within opposite sides. I remember that correctly. Yes. So now that we've gone from a cis to a trans configuration, that's basically when this cascade happens. This opposite molecule will activate transducing. Transducing activates phosphodiesterase. Activated phosphodiesterase turns cyclic GMP into GMP. And as we deplete the populations of cyclic GMP can no longer activate the sodium channel. So the sodium channel closes itself. It's no longer being activated by cyclic GMP. Now that it is closed, sodium cannot enter the cell. And that's the cell returns to its resting membrane potential, basically, of negative 70 millivolts. Whenever this occurs, neurotransmitter release decreases. And that decrease of neurotransmitter release, that is the signal that we somehow translate into images within our head. Whenever this transition from cis to trans retinal happens, now we have all of the retinal in a trans conformation. How are we going to activate it once again? It has to return to the cis conformation. The process of converting cis into trans repeatedly, that's called bleaching. And that's what happens whenever you stare at the sun and all of a sudden all you see is a ghost image of the sun. And that is because you have bleached all of your retinal into this trans conformation. It's all into trans conformation, and it cannot return to cis fast enough for it to be activated once again. But this is the usual process. Whenever the retinal gets bleached and it goes into a transformation, an ATP consuming enzyme will turn the retinal into the cis conformation, and then it once again forms with the opsin molecule, the rhodopsin molecule that's ready once again to be activated. Dark adapted, this is basically throughout the day there's some level of photo bleaching that occurs because we're constantly receiving signals. So whenever it starts getting dark, it takes some time for us to, completely turn all of these trans back into cis retinal. But once we do so, we're more akin to dark conditions. We're able to we adapt, and you intuitively feel this whenever you go from a bright room to a dark room. It's like it takes you some time to adapt to it. That's because there is level of photo bleaching that is occurring, and as we adapt, this retinal is it takes some time for the retinal to go from that trans to cis conformation that can readily collect photons or respond to photons of light. Okay, so we talked about convergence and divergence. So this is what's known as a receptive field. Here we have this basically this population of photoreceptors being innervated by a bipolar cell, and they all send a signal to it. So this bipolar cell will respond. Okay, so these blue photoreceptors being activated, these red photoreceptors are being activated. These populations are being activated, and it will respond or not depending on how many, depending on the wavelengths of light that come in. That, let's say we're just staring at a red square, then the blue photoreceptor is not going to be activated. So the possibility of that ganglions of that bipolar cell to be activated itself is going to be lower or higher, because it's kind of opposite, right? Right. Okay, we all are kind of aware that we have a circadian rhythm. There is so basically these photons of lights and the interaction of the pineal your pineal gland and photons and the visual system eventually will send all together alongside the suprachiasmatic nucleus, will in turn affect the function of the brainstem. That makes sense, right? That's the brainstem is where the reticular activating system is. We talked about how in the medulla oblongata, we have this system that basically is the starter of your brain, that starter signal that gets activated whenever you first wake up. That's your reticular activated system just coming into consciousness, restarting your brain. I mean, it is somewhat active. That's why we are able to perform the other functions. However, it's not active to that level of conscious behavior that we experience. Okay, so I don't know if this will help some of you. I hope it doesn't. I don't think it will hurt. I hope it helps. Some people say they're bad test takers, and those people are probably right. But it's not because you're just innately born with the inability to take a test. It's because we have only a limited amount of computing power that we can do simultaneously. And it's limited. We talk about probably the most intuitive way that we can understand our computing power on the now basis. Maybe we can do some amazing complex things. However, a lot of them are reflexive. We talk about a lot of them are a lot of them are reflexive. but the things that are new and that require immediate, like require computing power, such as like remembering a phone number, that's basically 9 digits long. That's like we only need to remember 9 pieces of information, and we can't. It seems that remembering 9 numbers is less complex than you know, your visual system, or just reading a word. It seems that in terms of computing power, remembering 9 digits is way less complex. But the fact that we cannot remember them, it should give you an idea of some of the limitations that we have. Whenever you're on a test, thinking that you're not on a test, that takes away some of that computing power. So if you had like 9, you're trying to remember a phone number, and you know you have 9 bytes of information that you need to collect, thinking that you're not good at test taking, that takes away 2 bytes of information. That takes that computing power. So now you only have, say, 7 bytes of information that you can process, because we spend that time thinking about this. So next time you're on test, if some of those thoughts come to mind, and say, fine, I'm bad, this is not the time for my brain to remind me of that. So just like focus on the task, keep all other thoughts away, because every time a thought that's not the test itself is intrusive, it takes away some of your computing power. Anyway, let that apply to test or life. And the reason why I was mentioning this is probably that's the reason why we sleep as well, because during sleep we perform memory consolidation, and being awake itself, or it's kind of consuming, or being conscious consumes some of that computing power of our brain. But if we're asleep, then we're focusing entirely on the memory consolidation portion of it. And that's probably one of the benefits of it. Okay, visual pathways. We talked about this is basically the left hemisphere is going to be mapped into the right hemisphere mapped into the left hemisphere. This is done through a combination of decussation of neurons going into the opposite hemisphere, and neurons staying within the same hemisphere as well, because each retina will receive fields from both hemispheres. But he would say, okay, some of that needs to go to the opposite to the opposite visual cortex, and some of that needs to say to the visual cortex within this hemisphere. And so those the neurons will either decussate, go to the other hemisphere, or stay within the same hemisphere. The end result of it, though, is the left visual field, whatever you're studying on your left portion, in this case, my lamp is going to end up in my, in my case, my lamp, which is my right hemisphere, is going to end up in my left visual cortex, and my screen that I'm always staring at instead of staring directly at you, that's on my left hemisphere. That's on my left visual field. That's gonna end up in my right cerebral cortex. How are we doing on time? Oh my, we have 4 minutes. Let's go through. Now I'm gonna go through the anatomy of the ear. We kind of saw this in the lab. So that we look at this, the one thing that I will remind you is like some of the physiology. Here's the tympanic membrane interacting with the ossicles themselves touching these windows here. We have here the oval window. That's where the stapes kind of sends its Morse code, its signal it disturbs the fluid within the cochlea and vestibule, and eventually, once it travels, it makes the pressure is released here in the round window. Okay, here's the inner ear. It contains endolymph. It contains all of these areas: the vestibule, the semicircular canal, and the cochlea. Vestibule will contain utricle and saccule, and semicircular canal will contain ducts within it. Okay. So here is, how are we doing? I'm going to go over some of the physiology of this during class. We still have, actually, let me just go over the physiology. I'm gonna go over the physiology, and if there is any question, we supplement during the lab. But some of the physiology, here are the hair cells within in this case, we have hair cells within the utricles. These hair cells, if the movement of the fluid is towards the kinesiology, and there would be an stimulator. It would be, the cell would be stimulated, releasing transmitters that would activate the sensory ending. If the signal is away from the kinesiology, on the other hand, that inhibits the signal, so it no longer fires. And this, what's being depicted here is all of this fluid and these particular structures, these crista and macula that contain these hair cells that allow it to sense the fluid, the movement of fluid, basically. See, these specialized stereocilia and kinesiology are there to sense the movement of fluid, and they have receptors within their membranes that respond accordingly. This is what we're going to see is these areas responsible for equilibrium. So the semicircular canals, you see their disposition, they're placed in an XYZ type of direction. So any movement will move fluid, specifically one of the semicircular canals. And that fluid eventually will interact with these structures called crista. And the structure called crista contain these hair cells. So the movement, depending on which direction it is, it would affect the different stereocilia differently. We also have within this area within the vestibule and the utricle, we have the presence of these structures called a saccule. So you will have this particular the crista responding to the semicircular canals. So these are the hair cells present here, the stereocilia and kinesiology. This is how these crista are disposed. You have sensory nerve hair cells, the stereocilia present here, and you have a cupula, gelatinous area, that's going to basically serve like a sail, so that fluid moving, it will be able to detect fluid movement more readily, and the hair cells will be here. So if this whole thing moves, then the hair cells kind of move. On the other hand, this is at the same level of the semicircular canal. The level of this structure called the saccule, we have these maculae. Maculae are going to be composed of this gelatinous material that contains on the surface this crystal, this carbon crystal, these calcium crystals called otoliths. These crystals are basically, they're sitting there, and by gravity they're affecting these crystals present here. Whenever there is movement, the crystals will move, or whenever you jump, the crystals will suspend within space briefly, and that lack of pressure within the gelatinous area will inform the hair cells that, hey, there is movement. This person just jumped. This is the movement of the semicircular canals in action. This is movement of these otoliths, these crystals, and putting different pressure against the gelatinous membrane. So this vestibular neural pathway involves cranial nerve number 8, the vestibulocochlear nerve. So far, we've only talked of the vestibular portion of the vestibulocochlear nerve. The cochlear portion, so this is the vestibular portion. You see this is the vestibulocochlear nerve present here. It's a vestibular branch that interacts with this crista and the maculae, and the cochlear branch, will see it interacts with the cochlea, and that's responsible for our sense of hearing. So this is a view of a cross section of the cochlea. Here is the cochlear portion of this nerve, and you see the cochlea will have this. It has this spiral area that is divided into these different ducts, these different cavities. You will have a vestibular duct and a tympanic duct. Fluid, here is the stapes bouncing against the round window that moves the fluid along the cochlea. It will go up through the vestibular duct and come back down through the cochlear duct. So it goes up through one of these cavities, and it makes it down through a different cavity. Where does it end up? It ends up by the round window. This connective tissue structure that basically is there to help dissipate the wave of fluid that just came down from the tip of the cochlea. So what else do we see here? We also have the cochlear duct present here. So it's basically 3 cavities. The main ones where the fluid is flowing through is going to be the vestibular and the cochlear cavities. We will also have these different membranes present. We're going to have a basilar membrane, a tectorial, and a vestibular membrane, all in relationship to these cavities. We're going to get a better view here. So this is one of those cavities, basically. We should understand them as these 3 different cavities that form a unit, okay? This is the vestibular duct, the tympanic duct, and the cochlear duct. Altogether, they form a unit, basically. They're going to be the vestibular duct, it interacts with the vestibular membrane, the tympanic duct with the basilar membrane, and then the basilar membrane through the hair cells will interact with the tectorial membrane. Here we have basically, this is the gist of our auditory sense. You have here the basilar membrane. You know, when fluid flows through here, or whenever there are disruptions between the basilar membrane, whenever there are disruptions between these cells and the tectorial membrane, they will respond accordingly through firing of releasing of molecules that will interact with these nerve fibers of the cochlear duct, of the cochlear nerve. So here it is. This is the gist of it. You have these hair cells. They interact with the tectorial membrane, and this movement, it's going to determine whether they'll be activated or not. These are just different properties of sound. The sound is just movement of air. Movement goes. This movement occurs in wavelength, in waves. Here's a definition of wavelength. Frequency. Pitch is going to be the difference between frequency. Amplitude is going to determine how loud. It's going to be how high these waves are. It's going to be determine how loud something is. And resonance are objects that vibrate at similar frequency. Pathway of sound, auditory ossicles apply pressure into the oval window that moves perilymph of the scala vestibuli. And these pressure waves distort the basilar membrane. On the way to the scala tympani, and this will allow the hair cells to be pushed against this tectorial membrane. And that interaction between hair cells and tectorial membrane, that's going to determine whether they get activated or not. So this is what we're depicting here. Sound waves arriving into the tympanic membrane that activates the ossicles activating the oval window, which activates the perilymph within the vestibular duct. This in turn affects the movement of fluid within these ducts, affects the basilar membrane, and the basilar membrane, it's where hair cells are going to be embedded, and those hair cells basically interact with the tectorial membrane. So they're going to basically move up and down against the tectorial membrane, and that movement, that kind of interaction, that physical touch, is going to open receptors within these hair cells that are going to trigger that movement, that signal. Eventually, this wavelength is going to return through the tympanic membrane. What we're observing here is how different waves, different frequencies affect different regions of the cochlea. High frequencies of 1600 hertz are going to mainly affect regions near the origin of the stimulation, and long and short frequencies are going to stimulate farther along the cochlea. So we send a signal. So whenever a signal arrives, it's being sent to the. So it's going to be sent to the inferior collicula of the midbrain. It's going to be sent here to the inferior collicula. It's going to be sent first to the superior olivary nucleus of the pons and the inferior collicula present here of inferior collicula of the midbrain. And in turn, this is going to be sent to the thalamus, and the thalamus is going to be sent to the primary auditory cortex within the temporal lobe. All right, that's it. We went over 11 minutes, but we were able to squeeze out the visual and auditory system within one of these sessions. I'm sure you have questions. This is the time to have them.
I do have questions if you have time to stay afterwards.
Yeah, you need to talk to me. I'll stick around. Any other questions or any other comments?
No, thank you.
No, thank you.
All right. Monica, I'm going to pause this recording so we can talk.
Okay, thank you.
Good night.
Good night, everybody.